Kia ora everyone, and welcome to this next mini lecture. And what we're going to be doing is wrapping up the mutualism section, and to do that I'm going to start to talk about lichens. So this mini lecture and the next one, which will be the final one, will both be about lichens. And lichens are really amazing things, and hopefully I'll be able to convince you of that and give you a bit of an insight into the nature of lichens. So I'll turn my video off and we'll start going through the slides. Okay, so to get started, so lichens, actually the origin of the word lichens is pretty unclear. So it comes from ancient Greek, but it's unclear which word it actually comes from. And there's two ideas. So one of them is an ancient Greek word for wart, and the other one is a word for suck up. And both of them kind of make sense. But anyway, it's a bit of a, as I say, it's a bit of a mystery where that name actually comes from. But I'm sure you've heard about lichens. And so lichens are a mutualistic symbiosis between a fungus and an alga, or occasionally between a fungus and a cyanobacterium. And they are incredible because they are really unique in the fact that their partnership forms a separate organism. It's completely kind of normal organism in terms of that if you didn't know that it was a symbiosis, you would just think it was a single organism. And the morphology of this organism is radically different to either of the partners that live alone. So I'm sure you've all seen lichens, you know what they look like. There's a huge variety of forms of lichens actually. But that ability for it to transform from two um, independent organisms that have their own morphologies to something that has a completely different morphology is, as far as I know, unique on Earth to lichens. And they're very successful, as we'll talk about in a minute. And so it's kind of amazing that there's been nothing else that has managed to develop this radical different morphology as a result of living in a symbiotic relationship. Now, I said that they're a mutualistic symbiosis. So what's the nature of the mutualism? What are the benefits that they get? So obviously, again, the fungus gets the carbohydrates, the sugars that are made by photosynthesis from the photosynthetic partner. And because sometimes the photosynthetic partner can be an alga and sometimes it can be a cyanobacterium, the general term for the photosynthetic partner of a lichen is a photobiont. And what does the photobiont get in return? So it's thought that the advantage it gets is protection, and we'll look at how that protection works in a minute. So going on to slide number 2, and so lichens are very highly diverse organisms, and we can, I guess, see this because more than 20% of all described fungal species, so in other words, about 14,000 different fungal species, form lichens. So they undergo this mutualistic relationship with algae or cyanobacteria and form different lichens. And so those 14,000, we recognize each of those as different lichen species, as we'll see in a minute. But curiously, there's very few photobiont species, so only about 100 photobiont species are known in total. And the kind of upshot of this is that lichens are named for their fungal partner, and usually they're highly or moderately selective for a photobiont partner. So in other words, a given fungus will form an association with a given type of alga or cyanobacterium to form a lichen. Whereas obviously because there's so few photobionts, alga will form, or cyanobacteria will form lichen associations with a large variety of different fungal species. So hopefully that kind of asymmetry in the relationship is clear. Now, one of the most surprising things about lichens is they're not just one group of fungi that have evolved this relationship. It appears that the lichen lifestyle has evolved independently several different times, despite the fact they have morphological and I guess functional similarities. And why do we think that? The reason we think that is that lichen fungi, you can find fungi that form lichens within the Ascomycetes and within the Basidiomycetes. And so it seems that they've evolved, these different groups of fungi within the fungal kingdom have evolved this lichen forming ability independently. We think it's an ancient association, and it's possible that the ancestor of all the fungi, of all the Ascomycete and Basidiomycete fungi, formed lichens, although I think that's probably unlikely, but we can't completely rule it out. But regardless, it seems to be an ancient association. So let's move to slide number 3 and look a little bit about the ecology of lichens. So I'm not going to say too much about this, but kind of amazingly, they're a dominant organism in about 8% of Earth's land area. So what I mean by that is about 8% of the land area on Earth, the dominant species present there is lichens. And there's some examples of lichens shown on the slide here, the photos. And so you can see that there's these kind of crustose lichens over here on the top right, and then these kind of beard-like lichens down here in the bottom right, and then these kind of amazing structures where the red bits are the sexually reproducing structures of the lichens here, so kind of almost tree-like lichens, even though these are quite small. Now, the reason that they are a dominant organism in such a large part of Earth's land area is that they're very adept at colonizing barren and dry environments. So they're extremely desiccation resistant, meaning that they can occupy environments that other organisms can't because of the lack of water. And you'll probably recognize that. You can find lichens that'll grow in rocks, as is shown on the picture on the right, and those kind of hostile environments. Kind of paradoxically, though, they're very sensitive to environmental pollutants, and so as such, they're used as environmental indicators to tell whether an environment has been degraded or not. Right, let's go to slide number 4. And what I want to talk about here is how lichens form and how they reproduce. So most of those about 100 species of photobionts can grow as free-living forms, either alga or cyanobacteria, so they can grow without their fungal partner. And similarly, most of the fungal partners can also grow as free-living forms. So you can take the fungus, separate it from the alga, and grow it, say, on a petri plate. But for most of these fungi, you don't actually find them out in nature growing free living. So whenever they grow, they end up forming lichen symbioses. Lichens sexually reproduce, but that sexual reproduction is not of the lichen itself, it's just the fungus. So the fungus will go through a sexual reproduction. It will produce spores, either ascospores or basidiospores, depending on whether it's an ascomycete or basidiomycete, obviously. And then those spores will germinate, and then that newly formed fungal individual will have to form a new association with an alga or cyanobacterium. And this process of how fungus finds its alga or cyanobacterial partner after it germinates is completely mysterious. So we don't understand how this happens. Did they just run into each other? Is there that many of these things floating around that they can do that? So really, we don't know too much about this at all. And the process is kind of diagrammed here on the picture on the right. Starting at the bottom, we have this is kind of symbolic of of the fungal hyphae, and here's single-celled alga, and they will somehow find each other, and then they will form these kind of primitive, disorganized-looking structures where the fungi and the algae are kind of coexisting, but they don't have a very clear morphology, and then that will somehow then grow up and develop into these things that have very highly specialized morphologies, such as these kind of cup-shaped lichens growing out of this lichen mass down here. And in this case, these cup-shaped structures are the reproductive structures, and again, they're the reproductive structures of the fungus itself. Even though they've got algae in them, the algae are not being reproduced through these. And so under slide number 5, so lichen formation and reproduction, continuing on with this. So as I kind of said, the initial development is quite disordered. So there's not really much in the way of organized morphology early on, but it then becomes very organized into what we call a lichen thallus. So the lichen thallus, this word you may have come across before in the course. In fact, I think I talked about it when I did Marge's lecture. It means a fungal individual. And so we've come across the term mycelium as a fungal individual. Thallus is the equivalent form, but of course in a lichen, it doesn't just contain hyphae, it also contains the alga. So the thallus is that individual. And this disorganized growth can be seen here when we have this picture on the left, which is a picture of things called soaridia. And so soaridia are the way that lichens can reproduce themselves in terms of the whole lichen being reproduced. as opposed to what I talked about before, which was the fungus reproducing itself within the lichen. And the way that this reproduction happens is really quite simple. So just by breaking bits of the lichen off, those little fragments of lichen can go and colonize somewhere else. And the bits that break off, the fancy name for those is Ceridia, as is indicated here. And on the left here, this picture shows the Ceridia, where there's just a few alga and a few hyphae that have this disorganized form, and then they will grow back up and form these highly specialized morphologies that we recognize as a mature lichen. And it's not just a random process, so a particular combination of photobiont and fungus will produce the same morphology time and time again. So it's a distinct morphology that's unique to that fungus photobiont combination. Okay, moving on to slide number 6. What I want to cover now is the structure of lichens. And so for most lichens, when we consider lichens, we often consider the macrolichens, so the ones that we can see. There are smaller ones, but for the purposes of this course, we're just going to consider the macrolichens. And so for these, the fungus builds the main lichen structure, the thallus, as I talked about before. And you can see that in the picture here. And so the white cells are the fungal hyphae, and the green cells obviously are the photobiont. And so the photobiont is positioned inside the lichen, so it's completely surrounded by the hyphae here. And here's another picture over here. You could ignore these letters, but again, the clear or the bluish bits and the brownish bits are the fungal hyphae, and the green bits are the photobiont. So the photobiont is inside the hyphae, and obviously to photosynthesize it requires light. So you can see that these hyphae are kind of transparent, and so that allows the light to come through. Furthermore, because these things undergo photosynthesis, and so they're fixing carbon and producing oxygen, you need to have gas exchange occurring within the lichen. And so that occurs through these pores, which is indicated by the P here. So that it can be gas exchanged through the lichen. And so this fungal-derived structure provides protection to the photobiont. And in particular, it provides protection against desiccation. And we'll look a little bit more at how that happens in one of the following slides. But that's the basic structure of a lichen, where the photobiont is encased inside a hyphal structure. Here's an electron micrograph of a lichen that shows in a bit more detail the structure, and we're going to introduce some basic terms. So there's the cortex and the medulla that we're going to think about. So the cortex is the surrounding layer, and so we have an upper cortex, which is the UC here, and this consists of densely woven layers of hyphae, and the lower cortex is the same. The photobiont, you can't really see them very well in here, but here's the photobionts in this layer here. And the medulla is the stuff in the middle, and this is loosely arranged hyphae that can be interspersed with the photobionts, and this is kind of the spongy layer. And cyphella down here, this is the fancy word for the pore that allows the gas exchange that I talked about. You'll also see this thing called T here. We're going to ignore that. So M is medulla and PH is photobiont, obviously. So again, just to show you in a bit more detail, the structure, and in particular the tightly woven hyphae of the cortex and the loosely woven hyphae of the medulla, and that's where the photobionts hang out. Okay, on to the next slide, slide number 8. One of the reasons that lichens are so desiccation resistant is that the fungi produce a layer of polysaccharides, and often this is quite a dense layer of polysaccharides in the outside, the peripheral part of the cortex. And this layer has hyphae, polysaccharides, and also a variety of secondary metabolites that are produced by the fungus embedded in it. And you can't really see much from these pictures here. I'm not expecting you to take much home from these, but these layers contribute to making the lichen very desiccation resistant. And the nature and the extent of this matrix of polysaccharide hyphae and secondary metabolites varies a lot between different lichens, and is one of the things that makes the lichen, gives the lichen its unique morphology. Okay, so I'm going to stop there, and in the final mini lecture, I'll continue to talk about lichens, and we're going to consider one particularly surprising, well, at least surprising for people who studied lichens, aspect that's been discovered recently about them. But for now, I'll stop. And if you have any questions, please direct them to Piazza.